Hello and welcome to this series of lectures on understanding bias and the introduction of variation as an evolutionary cause. Explanations in science and in everyday life often tell a story or follow a pattern, and an important explanatory paradigm in evolutionary biology is what the philosopher Elliot Sober calls equilibrium explanation, where an evolved trait is treated as the endpoint of a process of equilibration. So think about a solution of sodium chloride made with specific amounts of salt and water. This system has, when we add them together, the system has fast kinetics and it proceeds rapidly to its equilibrium state, regardless of the details. We could add the salt 1 grain at a time or all at once. We could stir it to the right or we could stir it to the left, and none of that would matter. The resulting solution would have predictable properties like a specific gravity or a boiling point that are predictable just from knowing what the components are without knowing the exact history. And we can explain or predict these properties because they represent this equilibrium state or attractor on which the system converges. Now, if a dynamic system hasn't converged like this, then in order to explain the current state, we need to know some prior state plus the intervening dynamics, the intervening kinetics. So in their famous defense of the Adaptationist Research Program in 1993, Reeve and Sherman explained that the observation that boulders collect in mountain valleys can be explained without knowing each boulder's precise trajectory. And likewise, evolutionary biologists can explain and predict observed phenotypic features without knowing the exact evolutionary trajectories, because in each case, a finite set of stable states serves as attractors for a large, and indeed they say infinite, number of possible histories. So this kind of explanatory paradigm is the perfect complement to the neo-Darwinian theory and to adaptationism more generally. And indeed, this paradigm is pre-evolutionary in the sense that creationists also sought to understand the species as the perfect finished endpoint of a process of creation, a process that goes to completion. Now, because of this importance of this paradigm, there was actually a struggle within evolutionary biology to establish the opposing notion of contingency. That is, evolved states cannot be explained entirely in ahistorical terms as attractors. Instead, in cases of contingency, the explanation requires some details about the starting conditions or the intervening dynamics. And one flavor of this contingency claim is what we might call mutational contingency, exemplified by cases like these, where the explanation for an outcome is that a specific mutation, or it depends on a specific mutation happening in the past, or an ancestral lineage that at some point in the past has the capacity to generate a particular kind of mutation at a high rate. So the citrate utilization story from the long-term evolution experiment, the PIDX1 deletion hotspot shown here, or the recent story about the role of an allo insertion in primate tail loss. So contingency in this sense is an explanatory concept. It's not a cause. There's no force of contingency. It's kind of a placeholder that indicates the negation of equilibration. So, and more generally, contingency, chance, and constraints are ways to negate this old paradigm of perfection without really replacing it. So they invoke an alternative paradigm or a counter paradigm of imperfection. And it makes one think of the Japanese philosophy of wabi-sabi, for instance, of finding beauty in imperfection, or the Japanese art of kintsugi, repairing pottery, broken pottery with gold. So the gold puts the focus on the cracks, the failures, which are treated as a unique and kind of endearing aspect of the object that tells its story. It makes it an object of nostalgia. And evolutionary biology, I think, is dominated by nostalgia. We can't get rid of old concepts. So we still have the equilibrium paradigm, but it's the broken kintsugi version with several large cracks representing chance, contingency, and constraints. But why focus on imperfection? What would evolutionary biology look like if we didn't have this kind of nostalgia? What if we just throw away the broken bowl and find a new paradigm that's more suitable for a world of propensities? Evolving systems have tendencies of change, one could say, and our job is to understand those tendencies and to predict and explain them without any need for a concept of perfection or imperfection. How would we represent evolutionary tendencies? What would be the quantitative and causal theory for how mutational tendencies influence evolutionary tendencies? What kind of evidence would we want to collect to sharpen our understanding of this dependency? If these are questions that interest you, then you're in the right place, because where mutational contingency ends, this project begins. And in today's talk, I'll start out with empirical results that indeed sharpen our understanding of the kinetic dependence of adaptive change on mutation. And I'll go through 5 different types of increasingly convincing or sharp evidence. And the simplest kind, corresponding to the simplest type of question one could ask, is whether there's a systematic enrichment of mutationally favored changes. Do we see more of what mutation favors? So what about transition transversion bias, which is a simple kind of nucleotide mutation bias? Out of the 12 different types of nucleotide mutations shown here, 8 of them, shown in red, are the between class, between purines and pyrimidines, transversions, and 4 are the within class transitions. So the background expectation, just based on the numbers, is 1 to 2. But there is a bias in individual rates, so the blue rates tend to be several-fold higher than the red ones. My colleague David McCandlish and I looked for an effect of this bias in parallel adaptive changes. So when we're considering the distribution of transitions and transversions among these changes, we can count up. So this is a small table here showing a few changes. So we can count up either the paths or events. So there's 4 different events. Each was a specific evolutionary change that took place in a particular species. But those 4 events happen along 3 different paths. The middle 2 of them here happen along the same path. So 2 events along the same path are parallel events. And when we're looking for a bias, then formally we're just looking for kind of a one-sided effect, assigned effect relative to a null model. And the null model is that the outcomes are determined by selection alone based on fitness effects, and regardless of which forms are more mutationally likely. So for transitions and transversions, each nucleotide has 1 transition and 2 transversions. So the null ratio that we can expect is a 1 to 2 ratio. So here's an experimental example of a transition bias. So in this experiment, Meyer and colleagues, so each row here is a strain and each column is a particular mutation associated with an adapted strain. So Meyer and colleagues carried out replicate adaptation of phage lambda. And again, each row is a strain and each column is a specific change in the phage J gene. The boxes indicate mutational changes found in adapted strains, with black for transitions and gray for transversions. The authors isolated 24 strains that had a new host specificity, and they compared those to 24 other adapted strains in the bottom block. So overall, you can see there's an obvious bias toward transitions. There's more black. When we just count the paths, the columns, there are 26 black ones and 12 gray ones. That's a 4-fold excess over the 1 to 2 null expectation. And when we count events, there's 191 to 48, which is about 7-fold above the null expectation. So in this meta-analysis that I mentioned, this study, along with 4 others, were just combined all together. So here's that lambda study. Here's another large study on FX174 that went through successive host reversals. And then there's several smaller experiments. And if we just sum these up by paths, 43 to 20, that's a 4-fold excess over the null expectation. And 304 to 85, that's about a 7-fold excess over the null expectation. So again, the point of this meta-analysis was just to gather enough data to do statistical testing. And the result is that there's a highly significant bias toward transitions in these cases of experimental evolution. Since that study was published in 2017, there's been additional studies that have also appeared. I'm just going to show these slides for those who are interested, but for the sake of time, I'm going to skip over them and not go into detail. So now let's move on from experimental evolution in the laboratory to unsupervised adaptation in nature. So here's one case. This is resistance to cardiac glycosides mediated by changes in the sodium pump ATP alpha 1. So that's targeted by some naturally occurring glycoside toxins, including both the cardiac glycosides produced by amphibians and the cardinalides produced by members of the dog pain family, and that's the focus of this chart here. So species such as the monarch butterfly have not only evolved sufficient resistance to eat these plants that produce this toxin, they actually sequester the toxin in their tissues, and that makes them noxious to predators. So this figure illustrates the data on ATP alpha 1 parallelisms reported by Jean and colleagues. And species that consume and sequester cardinalides are in yellow, and the ones that merely consume are in gray. And the bold columns are the experimentally verified effects. So in the next slide, I'll show the results of combining data on transitions and transversions from that study, as well as, or that case, as well as 9 other cases that include some famous examples among molecular evolutionists. So for instance, this represents spectral tuning of photoreceptors involved in color vision. This diagram represents some altitude adaptation in birds involving changes in hemoglobin, tetrodotoxin resistance. This is from a paper on parallelisms in ruminant ribonucleases, and this one is referring to parallelisms in echolocating bats and whales. So my colleague David McCandlish and I compared the combined results of 10 studies to a null model in which mutation biases have no effect. So again, the null is 1 to 2, and we see in terms of paths, what we see is about 1 to 1, so a 2-fold excess relative to that 1 to 2 expectation. And in terms of paths, about a 3-fold excess relative to the null expectation. So note for future purposes here that we're seeing a 2-fold or 3-fold effect, and these cases come from a variety of organisms, including animals and plants. Okay, as before, I'm going to skip over some slides with subsequent work in order to save time, and these slides are just here to show that this is an active area of research, and I want to draw attention to ongoing and new work that continues to provide evidence of the effect of mutation biases. Okay, so that transition and transversion analysis that I just showed you represents a simple kind of comparison looking for an excess of a favored class. So one way to go further than that would be to assess the quantitative influence of mutation on a spectrum or a distribution of changes, different types of changes. So suppose we're considering amino acid changes generally, just all of the missense changes. So if we have some set of adaptive changes, each of which is an amino acid change, they're often called missense changes. So each one has a starting codon and an ending codon for a different amino acid. So here's all of this ways that you can change a methionine codon to a different amino acid. And if we drew all of these paths, there would be 354 different missense mutations. So when we look at adaptive amino acid substitutions in some gene or genome, they're going to be distributed among 354 types. And we can write down a descriptive equation for their dependence on mutation to this, their dependence on the starting codon and some rate. And from this, we could just define a coefficient beta that captures the effect of mutation rates. So this is what is done in a new paper from Cano and colleagues that I was fortunate to be a part of. So an inferred value of beta of 0 indicates there's no influence of mutation, and beta equals 1 indicates a proportional influence. And we expect from prior work to see that range basically from 0 to 1. So this is a useful way to define mutational influence because prior work suggests that beta is going to range from a high of 1 under the limiting condition of rare mutation down to nearly 0 as competition among segregating alleles becomes intense. And in this new study, Cano and colleagues actually provide computer simulations that verify their approach. So the simulations generated distributions of adaptive substitutions in a model gene, and when the mutation supply is low here on the left, beta converges on, there's quite a lot of scatter here, but this is the central tendency of this line. So as mutation supply gets very small, beta converges on 1, and as it gets larger, it drops down towards 0. So these simulations provide some confidence that beta can be inferred from realistic data and that the inferred value tells us something about population genetics. So the authors take this, I'm sorry for this glitch over here in this figure. So the authors apply this method of analysis to 3 different sets of data on adaptive substitutions, including natural adaptation of Mycobacterium tuberculosis to antibiotics, and laboratory adaptation of E. coli to temperature stress, and laboratory adaptation of yeast to several different conditions from several different studies. So in this analysis, there's going to be an independent source of information on the mutation rates used to estimate this mu values for mutation, independent from this data on adaptive substitutions from these different experiments or natural clinical studies. So the result of applying this method is that in each case, the inferred value of beta is close to 1. The confidence interval, or the standard error, overlaps with 1, and it's significantly different from 0. So, and this is not just an effect of transition transversion bias, it's the entire distribution of rates for the 6 different types of nucleotide substitutions applied to all the missense changes. So, for instance, the species-specific mutation spectra for nucleotide mutations based on independent data did much better than randomly generated mutation spectra. That's what this top row of figures shows. And if you just infer the nucleotide mutation rates from the adaptive substitutions, that looks a lot like the empirically determined mutation rates from mutation accumulation studies. That's what's shown in this bottom series of graphs. So this recent work takes us quite far beyond the mere notion of mutational contingency. Instead, these results show that the process of adaptation sometimes results in a detailed quantitative correspondence with different mutational categories depending on their rates. So we don't yet have a really clear empirical understanding of how this dependency is influenced by the conditions of adaptation in nature. So that's an area where theoretical work is kind of a little bit ahead of empirical work, although much more work is needed in both areas. Okay, and in the interest of time, again, I'll skip over some related work on the emergence of drug-resistant tumors in cancer. So, we've seen evidence for a predictable bias favoring transitions in a broad set of cases, and we've seen a narrower evidence for the influence of the nucleotide mutation spectrum on the spectrum of missense changes. What kind of empirical studies might go even further than this? So I can think of a couple kinds. One way to go further from this is to actually manipulate the mutation spectrum. I'll talk about that in a moment. Another way is to focus on the rates of specific genomic changes rather than lumping things together into mutational classes. So an example of this second type would be a study by McLean and colleagues where they evolved resistance to the antibiotic rifampicin in many replicate cultures of Pseudomonas aeruginosa. Now, the resistant strains typically have a mutation in the RPOB gene that encodes RNA polymerase. So the authors provided mutation rate estimates for 11 mutants. I'm going to focus on that set of 11. And the mutation rates show about a 50-fold range of mutation rates, even though they're all single nucleotide mutations in the same gene. And they also measured fitness. So we have, for these 11 variants, the frequency of evolution, the mutation rate, and the fitness effect. And the relationships of those 3 variables are shown here. So we see right away in this second figure that the frequency evolved, the number of times a variant emerges and rises to high frequency in a replicate culture, is strongly correlated with the mutation rate. But that's not because, and fitness doesn't have very much effect because these are all strongly beneficial mutations. So there's not a lot of difference in the chance of fixation. And this difference doesn't have anything to do, so mutation rate doesn't really relate to fitness. So that's a further kind of evidence that's even more detailed for this close correspondence between the chances of adaptive changes and the rate of mutational origination. So a second kind of evidence that I think is even more powerful than like this kind of empirical mutation spectrum analysis that I showed earlier is to manipulate the mutation spectrum. I mean, that's really the gold standard to show that X causes Y, is to manipulate X and look at the effect on Y. So Alex Cousset and colleagues in this study subjected replicate lines of E. coli to increasing concentrations of the antibiotic cefotaxime, and they were using 3 different parental strains. There was a wild-type strain, there was a mut-H strain, and there was a mut-T strain, and these mutator strains have elevated rates of mutation, and more importantly, they have different mutation spectra. And the results shown here, these are the resistant strains that arose. The mutants are distributed among 3 boxes depending on the type of mutation, with the colors that indicate the parent. So the black bars indicate wild type, the blue ones indicate the mut-T, and the red ones indicate mut-H. So the kind of mutations that are favored by mut-T, AT to CG transversions, are shown in the left box. All right? And the center box shows transitions which are favored by mut-H, and the right box shows other kinds of changes. So because resistant strains derived from mut-T are shown in blue, and this box has the type of mutation favored by the blue, the mut-H mutator allele, the concentration of blue in this box is an indication that the mutation spectrum is affecting the spectrum of adaptive changes. And likewise, the concentration of red in this box. So again, this is kind of the gold standard to establish a cause-effect relationship. We directly manipulate the cause variable and show an influence on the effect variable. Okay, finally, there's an interesting experiment that sort of puts these two approaches together and considers the possibility of manipulating the rate of an individual mutation. So James Horton and colleagues observed in the very recent work that just came out that two different strains of Pseudomonas fluorescens adapt to the loss of motility in different ways. In one strain, over 95% of the time, adaptation involves changes in the NTRB locus, whereas in the other strain, various genes are implicated. And they identified a hotspot mutation associated with synonymous sequence differences in the two strains. And to test that this mutational hotspot causes the difference in the pattern of adaptation, they used genetic engineering to create the hotspot in one strain and to remove it in the other without changing the protein sequence, because these are synonymous changes. So here's the result. So when the AR2 strain is the one that keeps adapting in exactly the same way with the same mutation, this A289C mutation, either in rich medium or in minimal medium. But if you remove the hotspot, now we see a whole bunch of different ways that it adapts. And likewise, for the PF0-2X strain, which lacks the hotspot, it adapts by many, many different types of changes, whether in rich medium or minimal medium. But if you engineer the hotspot, then it shows this pattern of relying strongly on the A289C mutation. Okay, now that finishes my presentation of the evidence. Let's just go back and review it in two ways. First, in regard to establishing causation, and then second, in regard to that issue of explanatory paradigms that I mentioned. So when considering this theory that X causes Y, our first interest is often to establish if the proposed type of causation is ever real. Perhaps it's theoretically possible, but is there a known case? Is there any evidence that compels us, beyond any reasonable doubt, to accept that X causes Y? Again, I think the answer is yes here. The gold standard is to manipulate X under controlled conditions and show the expected effect on Y, and we clearly have that kind of evidence. So this kind of causation is real. What's the scale of this kind of effect? Well, it's clearly that a several-fold bias can impose a several-fold effect. We know this can happen from simulations, from theoretical work, and we also see cases where it's happening in nature or in experiments. So that's a substantial effect. What about the importance in terms of the scope? So, the experiments that most strongly demonstrate causal agency aren't the ones that show the broadest scope, because they're the experimental manipulation ones. What's the importance in nature? Well, it's the meta-analysis of transition-transversion bias that is the most important for addressing this issue of how widely do I expect to see this effect. Because that meta-analysis is based on cases that are listed in reviews and textbooks as examples of natural adaptation traced to the molecular level. These are exactly the kinds of cases that evolutionary biologists themselves consider to be important, and they come from animals and plants and microbes. So if we're asking this practical question, do I, as a researcher, need to consider mutational effects when I'm studying cases of adaptation like these? Well, the answer is yes, you do. Okay, what about this issue of paradigms that was raised at the start of the talk? So this table covers the set of 11 variants that I talked about previously from McLean and colleagues, where they measured, mutation rates were measured and fitness effects in this study of rifampicin resistance. So together, these 11 variants evolved 108 times, emerging and achieving high frequency in a replicate culture. So to apply the equilibrium paradigm, which applies to token outcomes, we would construct 108 stories, one for each outcome. And each story would say that selection chose the best variant from abundant variation. But the story would only be true in one case, because A1562T, that's the first one. These are ranked by fitness. That only occurred once. So there are 107 cases where a variant that we know that was more fit was not the one that was established. So obviously this paradigm of perfection, this equilibrium paradigm, is inadequate because evolution is not deterministic. It does not proceed inevitably to an attractor from infinitely many starting points. Now let's apply the paradigm of imperfection due to chance, contingency, and constraints. Again, this is a paradigm to explain token outcomes, individual instances. So we would construct 108 stories. And each story would say that selection causes rifampicin resistance by elevating the frequency of a beneficial allele. But the final state of the system, including its precise fitness, is contingent on the identity of that beneficial allele, which was not necessarily optimal and which could not be predicted because its presence in the initial gene pool was a matter of chance. Okay, it's contingent on the chance occurrence of some beneficial allele. So for instance, if we were to tell a story about A1562G here, we would say that we would tell this story 35 times, that selection caused adaptation by driving this allele to high frequency, but the identity of this allele could not be predicted because it was a matter of chance. Now, obviously, this paradigm of imperfection due to chance, contingency, and constraints is also inadequate. For instance, in these cases of A1553T, we don't know that this was the only variant that was present in the initial population. The other variants could have arisen and been lost. I mean, we could construct a more complex explanation in which we say, well, there could have been multiple alleles present, and A1553T was the one that happened to succeed and to reach a high frequency by chance. But these references to chance and mutation, they still fail to say what we need to explain, what we already know about how the world works. We already know this. Because in fact, the influence of mutation is not just a matter of chance. We see predictable regularities. So for instance, compare these two A1562T and A1562G that I mentioned before. So one of them, they are not just independent instances of evolution, they're alternative outcomes of the same conditions. They happen at different frequencies. And we know why. We can account for this difference. So A1562G is the one with the highest mutation rate. It's not the one with the highest fitness. And it's found 35 times, whereas A1562T is only found once, even though it has the highest fitness. And we can explain this. We know what's going on here. It's mutational origination. There's like a 30-fold higher mutation rate to A1562G, but the probability of fixation for this one is only slightly higher because they're both close to 1. So the kind of explanatory paradigm that we need to accommodate such factors, it's already in common use in molecular evolution. I would call it the Dayhoff paradigm. I'll say more about this in a later talk. So rather than pointing to evolved features and treating them as endpoints, we construct a quantitative description of the tendencies of evolutionary change, like a Dayhoff matrix of amino acid replaceability. And we confront that by asking questions about why one kind of change is more likely than another kind of change. Why do we see this kind of change more likely than this other kind? Why do we see transitions more often than transversions? Why do we see A1562G more than A1562T? So again, we're looking at like a matrix of replaceability or a spectrum of tendencies, and we attempt to explain why one kind of change is more likely than another by pointing to aspects of mutation, genetic encoding, and selection. So at the beginning of this talk, I criticized the equilibrium paradigm and the counter paradigm of imperfection, and I said, this project begins where mutational contingency ends. So now we can give this statement a more precise meaning. So empirically, examples of mutational contingency are really just an inducement or a starting point for further empirical work to explore more deeply the precise kinetic dependence of adaptation on mutation. So today, in 2021, we have a body of evidence that sharpens our understanding of this dependence far beyond what is shown by examples of mutational contingency. For instance, we can state that there are some cases that the coefficient of mutational influence, beta, is roughly 1. Secondly, we can consider mutational contingency as an application of the paradigm of imperfection due to chance, contingency, and constraints. And again, today in 2021, we can argue definitively that this paradigm is inadequate. We have a basis to prefer a different paradigm that's more suitable for evolution as a process that exhibits propensities that are predictable and which in some cases are due to mutational tendencies. And finally, we have a causal theory that will be discussed later in the series of talks. So contingency is an explanatory concept, not a causal force. However, there's a body of theory on the causal role of biases in origination, a topic that will be discussed subsequently. Okay, thanks for listening to this talk. In the next one, I'm going to go back over the evidence more critically to address some possible pitfalls and objections. This talk will be particularly important if you're one of those people who wants to get involved in this kind of research. Maybe not so important for the rest of you. You could probably skip this and go on to the next 3 lectures, which are about this causal theory that I talked about, explaining it and then putting it in the context of formal and informal reasoning about the role of variation in evolution. So if you have questions or comments, feel free to leave them in the comments section or find me online. I will now silently scroll through the bibliography for those who are interested in sources. Thanks again for listening.